Hello, I'm Sadhna, and today I will be talking about the complement system. Complement system consists of the series of proteins which are known as the complement proteins, which are working together to augment, to improve, or to, we can rather say, complement the activity of the immune system. So complement system has three main pathways. The first pathway is the classical pathway, the other is the alternate, and the one which has recently been discovered is the menin-lectin pathway. Now I was talking about the complement proteins. Complement proteins are like any other proteins in our body. They are synthesized from the liver, and but there are exceptions. For example, with complement protein number 1, which is synthesized from the GI mucosa, and there are others which are also synthesized from the macrophages. Okay, so we know that whenever a microbe attacks our cell, it enters into our body, there is inflammation, and as a result of inflammation, there is the endothelial lining which has been affected, and the gaps are created between the endothelial cells, and most of the proteins which are already there in the blood, they are seeping out of the endothelial lining. So meanwhile, what happens is, like for example, we take a microbe. There is the antigens which are expressed on the surface of every microbes, and these antigens, we have the antibodies in our bodies which are, for example, IgG or IgM, which are very specific for these microbial antigens. So what it will do is, when these microbes will be there in our body, these already inactivated, already present antibodies which are there in our blood will be going towards that microbe and form a complex with the microbial antigen. So if we can have a look at the diagram, it will be more clear. For example, this is the microbe, and these are the surface antigens on its surface. We have the antibody, and this is the Fc end of the antibody. So what happens is that this antibody makes a complex with the microbial antigen, and it gets activated. So once it gets activated, it will attract the complement proteins. This is the endothelial cell blood vessel, and these are the cells which are having the gaps, which are even more wide when there is inflammation, and more amount of proteins and blood will be seeping out. So the first protein which will be getting attached to this complex is the C1. Now the C1 will activate the other protein, which is the C4, and the C4 will be hydrolyzed into its component C4A and C4B. So the C4B remains attached with the C1, and this C4 will go away. This C4B will again activate another protein, C2B. Now you will have a confusion that why C4 is coming first and C2 is coming later. The reason behind this is very simple, that the C4 protein was firstly discovered, then the C2. So it is named or on the basis of the sequence of their discoveries. Now this C2B will activate the third complement protein, which is the C3. Again, the C3B complement will be staying with the C2B, and the C3A component will be going away. Also the C2A component similarly. Now this C4B and C2B will be actually together, they are activating this C3. That's why we can say that this C4 and C2 are the C3 convertase. Why? Because they are activating the C3. Now this C3b will be activating the C5 molecule, the C5 protein, and again the C5b will be staying back and C5a will go away. Now this C5b is being converted by the complex of C4b, C2b and C3b. So C4b, C2b and C3b can be also referred to as the C5 convertase. Right? Now after this whole protein series is being activated, this C5b will activate C6, C7, C8 and C9. Now C5b, C6, 7, 8 and 9 together will be forming a pentamer molecule. Okay, for example like this. You can say that this is C5a, this is C6, C7, C8 and C9, and this is known as the membrane attacking complex. Okay, this is the membrane attack complex. Why? Because this will be actually attacking the microbial membrane. They will make pores into the microbial membrane. Okay, so this C3b is actually the central complement protein. Why? Because it acts as an opsonin as well. As we can see that there is another pathway in which the antibody doesn't take the help of the complement proteins and it does all the action on its own. For example, this is the microbe, again the antigens, and this is the antibody. Now this complex will be directly presented to the phagocytic molecules or the phagocytic cells, for example the macrophages or the neutrophils. If this antigen is holding this microbe on one end and this is holding or making a bond or a complex with the phagocytic molecule on the other end with this receptor, which is the Fc receptor, because this receptor is binding with the Fc portion of the antibody. So now here the antibody is acting as a opsonin molecule, right? Because it is initiating the opsonization process, means it is preparing this microbe to be phagocytized by the macrophage or the neutrophil. Alright, now this is known as the direct opsonization, because we are not involving any kind of a complement proteins. So obviously if you are not involving the complement series, this has to be the fast method as compared to the complement protein pathway. Okay, so this is the now there is another way by which the microbes can be presented to the phagocytic cells. We take a microbe again, the antigen, and now this is having certain molecules in which the there is endotoxins. Endotoxins are the lipopolysaccharide type molecules which are present on the membrane or present in the membrane of the gram-negative bacteria. Because they are having, gram-negative bacteria is having what? It is having an extra lipopolysaccharide layer on the outermost, as an outermost layer. Okay, so these endotoxins will be actually attracting the C3 molecules directly. Okay, C3A will be going away, and this C3 will be combining with these endotoxin and forming a complex. Now this C3b will again activate another protein which is known as the CB, which is known as the BB complement protein, and this BA component will be going away. Now this C3b and the BB, this thing will activate, as a complex will activate another protein which is the C3b. Hence this can be also known as the C3 convertase. Alright. Now again this will activate the C5b, then C6, C7, C8 and C9. So the last or the terminal steps remains the same. Only the initial steps are different. Here we need the antibody. Here we don't need the antibody. Right? So we can say that this is the alternate pathway. Okay, so we have discussed two pathways. One is the classical pathway. This is classical pathway. Why? Because it is involving the antibodies. Here there is no antibodies involved. That's why we are saying it as an alternate pathway. Right? Now the recently discovered pathway, which is the Menin-Lactin pathway. What this pathway will involve, that there are certain microbes which generally gram-positive bacteria, okay, which are having, say, a molecule mannose in their cell membrane. Again, there will be, during the inflammation, the endothelial cell gaps will increase, and there will be lot of proteins coming out along with the other proteins and the plasma. So there is a membrane binding lactin, which is a protein which is already there flowing in the blood, but it seeps out in the case of during the inflammation. There will be a complex between these two, right? And this will activate C4 complement protein. It will get hydrolyzed. That means it will separate into two components. Similarly, C4a will be going away, and C4b will be binding to this complex. As we have already discussed, you can easily get an idea from here. The rest of the process remains the same, right? That means C2b, then this will activate the C3 complement protein. This will activate the C5 complement protein, then C6, C7, 8 and 9, and this will act as a membrane attack complex, attacking the membrane of the microbe. Okay, so this is known as the Menin-Lactin pathway because in this case it involves no antibodies. That's why this is again a type of alternate pathway. So we can say these two pathways are known as the alternate pathway, and this is the classical pathway, and this has to be the first line of defense. Why? Because we don't have any time when the bacteria or any microbe will attack our body system. We don't have much time. We can't wait for the antibodies to get activated and then help or defend us. So these two pathways will be acted in those cases as the first line of defense. Also, as I can say you, I have already talked about the opsonin molecules. The one opsonin molecules is the C3 protein, right? C3 protein, which I have not discussed. Let me discuss this with you. We have already mentioned that one opsonin molecule is the IgG, okay, or the IgM. So this IgG is acting as an opsonin molecule. So we can say this is the direct opsonization, right? But sometimes what happens is there will be the similar initial pathway. A microbe will be there, antigen will be there on the membranes, the antibody will be forming a complex, then the C1 will be activated, C4b will be there, C2b and C3b. That's it, up to here, right? So this initial pathway is now actually behaving as the complex which has to be presented to the phagocytic cell, for example the macrophages or the neutrophils. Okay, so now we can say that this C3 is basically the opsonin. Why? Because on one hand it is having the complex, which is the C3 convertase, antibody and the antigen of the microbe, and on the other hand it has binding itself with the C3b receptor, and this C3 receptor is on what? On the phagocytic cell, which can be either macrophage or neutrophils, right? So we can see in this picture so far that the classical pathway is involving the antibodies, right? And the alternate pathway are of again two types. There are the endotoxin involved pathway, which are having no antibodies involved, and the menin-lectin pathway. Now, in the classical pathway, these two molecules are acting as the opsonin. One is the antibody itself. It is directly attaching with the neutrophils or the macrophages. That's why it is the direct opsonization. And now this C3 is actually having antibody also, and this whole complex is actually presented to the phagocytic cell. That's why we can say this is the indirect opsonization, right? Indirect opsonization, right? And this endotoxin, this pathway, alternate pathway, is more commonly seen in the gram negative, because in gram positive, this indirect opsonization will do the work, right? They will do the defense against this microbe. But the problem is the gram negative bacteria. As I already told you that they are having a lipopolysaccharide in their membranes, right? So we have to have some other attacking complex which will make a pore in the membrane. That's why this particular last or terminal part is very important in the case of gram negative. So in certain people which are having congenital defect of deficiency of these type of proteins, they will be having they will be very vulnerable to the gram negative bacteria, right? Okay, so this was all about the types of complement systems. Now we can have a question here that why our host cells are not being attacked by these complement proteins, which are all the time, you know, circulating in our body cells, in the blood, because there are some molecules which are the complement cutters. Okay, these are the complement cutters which will cut or the complements if they are attacking our own host cell. Okay, these are the decay accelerators. Okay, so these are the cluster of differentiation molecules, CD55 and CD59. Okay, so this was the ones, and yes, this classical pathway is basically the acquired. Okay, so this classical complement pathway can also be referred as the acquired immunity. On the contrary, the alternate pathway can be referred as the innate immunity. Now, the last thing which I need to mention is the role of these metabolites of the complement proteins, which I have already earlier discussed, that these are the byproducts of the hydrolysis of the complement protein. For example, C2A, C4A, C3A and C5A. Now these also play a very important role. For example, if you take this is the endothelial lining, and these are the mast cells which are all over the body, lining the parallel to the endothelial linings. So these mast cells are having the C3A and C5A receptors. So these C3 and C5A receptors, which are actually the result of the complement system, these will be binding to these receptors, and this whole complex will be actually activating the mast cells. Now these mast cells will be activated and they will start secreting more amount of histamine, which will cause what? Which will cause more amount of this gap creation between the alloendothelial cells. And these gaps, if widen, this will pull out more amount of complement proteins, right, to the site of inflammation. So this will even more increase the effect of the complement system. So this is all what I need to share with you regarding the complement system. Thank you so much.